This is, by the way, our last seminar for the year. I wouldn't say we saved the best for last because they all were best, but this is a very good one. And I'm very happy to, after many years of knowing Hugues Abriel, to host him here at Washington University. Professor Abriel is the director of clinical research at the University of Bern in Switzerland. Those of you who know the University of Bern has a very long and distinguished history in cardiac electrophysiology, starting really with Silvio Weidmann, who was the first to record an action potential from a cardiomyocyte, and then developed the whole concept of gap junctions and conduction between cells, and I can go on and on, really remarkable. And that continued with the next generation of people in that laboratory and the Department of Physiology. Hugues received his both MD and Ph.D. from the University of Lausanne, so he studied in French, and now he's working in Swiss Deutsch. It's a big transition, and doing some beautiful work on ion channels, channelopathies, the basis for channel diseases, and really complex views of regulation of ion channels, in particular the sodium channel. I forgot to say that the first time we met was when he was at Columbia University working in Rocky Cass's lab. So thank you for coming.
Well, Joram, Dean, colleagues, new colleagues, future colleagues maybe, I'd like to thank you a lot for inviting me here in St. Louis. Well, it's indeed the first time I'm here, some sort of middle of the United States. It's almost as green as Switzerland, as you can see here.
Nice mountains.
Yes, we have mountains indeed. Where I'm spending or trying to spend as many weekends as possible. As you may see here, so a few coves. Those are very encoded are completely black. And these mountains, interesting is, it's when you're on the top of that mountain, you are, I think you would call it, biophysicist or a singular point, because there you would have to have 3 feet actually. One in France, one in Italy, and one in Switzerland. So yes, I'm not here to tell you too much about my weekends, but more about my working days at the University of Bern. So maybe first thing, well, for me, I just checked on my watch. It's midnight. I had a very long flight yesterday, so please apologize. Maybe sometimes I have to find my own words, but I'll do my best. And if you have a question, so don't hesitate to interrupt me. It's about these cardiac sodium channels, ADP1.5, and as Joram said, interacting proteins playing a role in regulating the activity of these channels and somehow the roles in cardiac channelopathies. I'd like to start always, these talks about ion channels, stating this fact, which is some sort of a paradox, right? Rare diseases are in fact frequent, and indeed, in the general population, there are about 5 to 10% of the population, general population, afflicted with rare disease. Most of the time, those are genetic disorders. And why I'm saying this is because we, in the field of ion channel physiology and pathophysiology, we have been able to learn a lot, and publish a lot, and learn a lot about the physiology by studying those many rare diseases. In fact, here we are talking about rare diseases modifying the function of ion channels. And you know that these ion channels are very important in most of cells of the body. Sometimes they are, allowing positive charge flowing into the cells or positive charge flowing out of the cells. There are also intracellular ion channels, as you know, But these membrane ion channels, when they behave wrong, so they may lead to problems such as cardiac arrhythmias. As shown here, this is a typical monomorphic, actually polymorphic ventricular tachycardia called dorsal de pointe. And this patient has a very specific type of channelopathy. In fact, this is not a genetic type of channelopathy. This patient has taken a drug called methadone, and this blocks very potently the hERG channel important for the cardiac repolarization. So one way to try to make some sense in this field of channelopathies, well, disease, human disease caused by malfunction of ion channels, is to use the classifications that doctors like to have within inherited or primary disease as compared to the one that are acquired or secondary, right? And if you want to classify channelopathies according to these distinctions, so you have the genetic ones, and most of the mutations that have been found in the past have been found in genes coding for either the ion channel, pore-forming subunits, or regulatory subunits. Those are the channelopathies that we have been studying most time over the past years. So, but in fact, most likely, in terms of the frequency, clinically, most of the problem arises with acquired or secondary channelopathies, which are caused by, as I mentioned at the beginning, when the patient is taking a drug and blocking or modifying the function of an ion channel. Sometimes those are autoimmune disorders, Graves' disease, or sometimes AV block. Pediatric cardiologist knows that very well in young patients. These may be also disease dependent in the context of a chronic disease, such as, for instance, well known for the cardiac field, heart failure, chronic heart failure will modify the expression and the function of many different ion channels, and this can be seen as an acquired channelopathies, and there may be other causes here. Cardiac channelopathies, I'd like to make a brief reminder about the basic aspects of cellular electrophysiology. And what we know, and as you said it before, Yoram, I mean, the first action potential of a ventricular cell has been recorded in Bern, and this was not very different from this action potential that is mimicking or schematizing the action potential of a ventricular human cell. We have resting membrane potential, depolarization very fast, early repolarization plateau, and repolarization, late repolarization. And what we know today, we have been able to dissect the different currents that are important for the depolarization, so our positive charge flowing into the cell, depolarizing the cells, or for the repolarization, positive charges flowing out of the cells, repolarizing cardiac cells. And what we know now is that those different currents, well, the red are shapes of these different depolarizing currents, sodium current, calcium current. We know now the molecular correlates, the identity of these all different ion channels. And we think that we have almost a complete, it's not the case, I think we are still missing a few important ion channels involved in the shaping and the generation of the cardiac action potentials. So we have these different voltage-gated sodium and calcium channels, and I will mainly speak about these voltage-gated sodium channel called NEV1.5, but it's very important for this very rapid depolarization of the cardiac action potential. And then we have different currents that are these repolarizing currents that are important for the early and late repolarization phase of the cardiac action potential. And here we have the different ion channels that are important for mediating these different currents and channels. This hERG channel, again, this is, for instance, the target of many of these different drugs that would lead to dorsal de pointe arrhythmias. So what we know also is, for the physiology, that the duration of the action potential duration is mainly the result of the resultant of depolarizing inward current and this repolarizing outward current. And if either you take a drug that will block the hERG channel, or there is a mutation in one of the genes coding for this IHL, so which may be a loss of function or loss expression mutation. This will lead to less repolarizing current, and by doing so, there will be a delay in the repolarization phase, and that will, this is the cellular cause of the long QT syndrome. Then, what we also know is that you can get a prolongation of the action potential relation if you have also an increase in inward currents, such as sodium and calcium current, and we'll come back to that in a minute. Nowadays, we also know that we may have disease not only when there is a prolongation of the action potential duration, but also when there is a shortening of the action potential duration. And this can be caused by, this is a very, a mirror image of what I just told you for the prolongations. So here it's maybe because it may be caused by a increase in this repolarizing current or decrease in depolarizing current, and will lead to short QT syndrome in patients. So this is a much more rare disorders than long QT syndrome. So nowadays, in 2016, the field of cardiac channelopathies became extremely complex, mainly because, there are many different genes, many different mechanisms involved, and here those are the different abbreviations of the different phenotypes. So doctors like to have those nice classifications. Sometimes we see that this may be a bit arbitrary, but for the so-called long QT syndrome, we have at least 15 different types, which are defined by the different genes, 15 different genes that have been related or linked to the congenital long QT syndrome, 6 genes for the short QT syndrome, 17 genes that have been linked. I say linked because I don't like the word cause, because in fact this is not completely clear whether they are really causal. They play a role in disease. To Brugada syndrome, ventricular tachycardia, 4 genes, conduction disease, 5 genes, 6 sinus syndrome, atrial fibrillations, and most of the genes, as I told you, they code either for the IHL subunit, the pore-forming subunit, or we call them also the alpha subunit, or for interacting subunits that are maybe the so-called beta subunits, or interacting proteins as IHL interacting proteins, and we will see in my talk. I've been mainly interested in these different proteins because the regulation of this pore-forming subunit has been shown to be very important also in disease context in humans. So when I started to be as a sort of young electrophysiologist, because I'm a physiologist, I'm not a geneticist at all, I'm interacting with many geneticists, and I have to learn many genetic concepts. I'm still learning every day about genetics, epigenetics nowadays. And so the fact when, I've been a doctor, working as a doctor, so we have a phenotype, we have a disease, we have a diagnostic, but what I had to learn is that for a given phenotype, for a given disease, such as the long QT syndrome, we may have several genes causing that disease. This is a bit problematic, but it's maybe a question of definition. So those are the different genes, KSCNQ1, KSCNH2, those are genes coding for the pore-forming subunit of K channel, potassium channels, and SCN5A is the genes coding for the sodium channel I just told you about, NB1.5. And luckily for medical genetics, most of the time, for the case of congenital QT syndrome, you would find mutations in one of the 3 different genes, and very rarely for this long list of genes shown here. So there is also the mirror image. Again, we have one gene, SCN5A, that is coding for NB1.5, and this gene may be mutated in a long list of phenotypes. It makes me things extremely complex and actually, in fact, very attractive trying to figure out, how can that be? Mutations in the genes coding for SN5A, which is coding for the sodium channel NB1.5, have been found in this long list of cardiac phenotypes. Most of them are purely electrical phenotypes, such as congenital long QT syndrome. We don't think that there is much morphologic alterations. But for some of these phenotypes, such as dilated cardiomyopathy, this is clearly the muscle phenotype that is striking here. We don't really understand how this is happening. And you see here this list making us trying to figure out how it can be, and we have this big bag of different phenotypes that we could call cardiac sodium current diseases. So this was the very general broad introduction about cardiac channelopathies, and now let me dig into the cardiac sodium channels, and mainly the work we have been doing over the past 10 years or so, trying to figure out what is a cardiac sodium channel in cardiac cells. And we came to the conclusion that, in fact, a cardiac sodium channel does not exist. There are several of them, and they are part of multi-protein complexes. So the cardiac sodium channels, if you look at the membrane topology of it, so that's how it looks like. We have, well, in fact, more than 2,000 amino acids, 2016 or 15, depending on the splice variants. And we have these domains, homologous domain 1, 2, 3, and 4. There are beta subunits. I'm not going to talk about any more about the beta subunits. And in each of these domains, we have 4, 5, 6 membrane segments. The 1 to 4 transmembrane segments are forming what we call the voltage-sensitive domains, and 5 and 6 are the pore-forming domain of the voltage-sensitive channels. But what we have been finding is that, in fact, they are interacting, and this is really schematically here described. There are many possibly interacting proteins. And for many of these proteins, we know exactly where they interact with which, most of the time, protein-protein interaction domains in the sequence of the cardiac sodium channels. For others, such as these proteins here, we don't know yet exactly how they interact with the cardiac sodium channels. They have been co-purified, co-precipitated, but they may be interacting via other partners. So I'm going to focus on mainly the proteins interacting with these last 3 amino acids, SRV, serine, isoleucine, and valine. And these 3 last amino acids are forming a so-called PDZ domain binding motif. And any protein that has a PDZ domain, which is a protein-protein interaction domain, may interact with these 3 last amino acids. And in fact, we found that at least 3 proteins are possibly interacting with this SIV domain. Citrofin, come back to that, which is a part of a macromolecular complex with dystrophin and maybe also utrophin. A protein called SAP97, I will talk about that. And another protein, which is a protein tyrosine phosphatase, shown here, PTPH1. Okay, so, but in fact, just by looking at this, because there is only one SIV domain, you will understand that since these interactions are exclusive, so you cannot have several proteins interacting with the same domain here. This led to the assumption that we may have different sodium channels, so some interacting with citrofin, some with SAP97, and some interacting with PTPH1. So this was a starting point to think, well, we have several or distinct multi-protein complexes of the cardiac sodium channel. I will show you the evidence that we obtained supporting this model. So I already told you this. So we have not one unique sodium channel. And for people like here, modeling the sodium channel, the sodium current, and mainly the regulation of sodium current, this is going to be important in the future. One will have to model that, for sure. But distinct pools or populations with distinct protein complexes. So that's one illustration, and that's one of the nicest illustrations that one can get so far of these different pools. This has been done by performing experiments using what we call this proximity ligation assay. This is done, people have developed this technique called also that co-immunoprecipitation in situ. So because you have two different antibodies. In fact, if you have two specific antibodies recognizing two different proteins that are either interacting together or very close together, and then you take these secondary antibodies that are being modified with oligonucleotides, and then you form such a PCR-like reactions with a dye. Everywhere where you have a red dot is where there is such a interaction or close proximity. And what we observed is that there is a population of NEV1.5 at the intercalated disc interacting with connexin 43, but also with plakophilin 2, as shown here. But then there is another one that is at the lateral membrane, and then here this is a population interacting with syntrophin, and I'll come back to these different observations with the different mouse models that have been generated. So, I'm going to briefly mention, how the whole project started in the lab a few years ago, and it has started with the fact that we observed that dystrophin and NEV1.5 were interacting together, and was a serendipitous observation. We were not looking at all at these interactions, but we stumbled upon this interaction. And I always say, when we've done that, that's one of the good reasons to do science and to stay into science, because you will find things that you were not looking for. And what we found was mainly these high molecular weight protein here that we found when we were performing so-called pulldown experiments. Those are fishing experiments or purification experiments, where we're using the C-terminus of NEV1.5, last 66 amino acids. And when we performed the same experiments, but when we truncated the last 3 amino acids, so we call it delta SIV because those 3 amino acids are missing, so we didn't observe that protein here. So this protein has been, by mass spectrometry, identified as being dystrophin. So it was not completely new. There was actually 1 or 2 papers suggesting that it would be the case, but what we have been doing here was to demonstrate the role of this interaction in myocytes. Well, first, what we observed is that while NEV1.5, the cardiac sodium channels, in sections of here, rat heart sections, seems to be localized not only at the intercalated disc, but also at what we call the lateral membrane. Dystrophin is only expressed at the lateral membrane. And where we have these 2 arrows here is where we have the NEV1.5 pool at the intercalated disc. You see that there is no dystrophin expressed. So when you do an overlap, you see that at the lateral membrane there is an overlap, so you have these yellow dots here, but at the intercalated disc there is only the sodium channel, it's just green. Similar with syntrophin. In fact, I have to say that we have been using here an antibody recognizing the different isoforms of syntrophin. There are maybe 3 of them expressed in cardiac cells, but here again you see that we have syntrophin expression at the lateral membrane, but not at the intercalated disc as shown here. So clearly we have 2 populations of sodium channels, the one at the intercalated disc and the one at the lateral membrane. So the model that we have is that the sodium channel at the lateral membrane is interacting with dystrophin via syntrophin, because syntrophin has a PDZ binding domain, and there is a known interaction between syntrophin and dystrophin, and it is part of these macromolecular complexes of glycoproteins found at the lateral membrane of cardiomyocytes. So what we could do at that time is to study the mice that are knocked out for dystrophin. This is a mouse model that has been developed since many, many years, the MDX mouse model. And what we observed is that when we looked at the expression of the sodium channel in sections of hearts from control to MDX, here it's a bit exaggerated. The contrast makes you think that there is a complete disappearance of the lateral membrane pool, but there is still a small expression, but clearly a strong reduction of the lateral membrane pool of the sodium channel in MDX mice. So then we continued, and since we had this lateral membrane pool, we tried to figure out, well, maybe we may find partners for the intercalated disc pool of sodium channels. And that's where it started a collaboration with colleagues from well, Germany and France, and here we have different types of Stephans, you know, the German Stephans, the French Stephans. So actually you could make this list even longer. And we were working on maggot proteins. Maggot proteins are extremely interesting proteins because they are adapter proteins with many different protein-protein interaction domains. And some of them are PDC domain, SH3 domains, and a very specific one in the kinase domain as shown here. And some of these proteins that have been very, very well described in the in neurons, and most of them are part of the synapses, were shown to be expressed in cardiac cells. And for a reason, I will not make a long story here, we focus mainly on SAP97, but I can tell you that in fact we are currently also working on another protein, which is the protein CASC, because both SAP97 and CASC are expressed in cardiac cells. CASC is well expressed in cardiac cells, and at that time, so well, I'll come back to where it is expressed in cardiac cells. But when we try to show, you know, interaction between SAP97, excuse me, and the NEB1.5 channels, so again we perform these pulldown assays with the last three amino acids or the truncated form as shown here, and clearly we could demonstrate that there was a specific interaction between the last three amino acids of NEB1.5 and SAP97 expressed or found in mouse ventricles or human atria, as you can observe here. Just mutating the last three amino acids abolished this interaction. So here the localization of SAP97, sorry. So SAP97 seems to be, at least in these rat sections, to be majorly expressed at the intercalated disc, and very little at the lateral membrane. In fact, we are revising this because now with new antibodies and with better sections, the thicker section, I think we see that we also have lateral membrane expression. But here, at least, there is a nice co-localization between SAP97 and NEB1.5. At that time, we were trying to figure out what could be the role of SAP97 in regulating the expression of NEB1.5, and we done this experiment in HEC293 cells where we silence SAP97 because there is a very nice expression of SAP97 endogenously in those HEC293 cells, but also in rat atrial myocytes where it has been silenced. And in both cases, what we observed was a very strong, robust decrease in the expression of currents, so which was due, in fact, to a reduction of membrane expression of NEB1.5. So I make now a short break just to explain to you that this has been, well, it has been published in 2011 in Circ Research. This has been the motivation why we thought, well, we are going to validate and we will develop mouse model to validate this observation. And as you will see at the end of my talk, we were not able to validate that. Actually, the result that we obtained in vivo with mice were contradicting the role of SAP97 in vivo. Come back to that maybe in the discussion. So,
are these the bar graph is the peak current?
Yes. Yes, yes. So we now I'm going to show you these in vivo mouse models, mouse experiment that we performed, trying to challenge or to demonstrate this model where we have lateral membrane NEB1.5 interacting with syntrophin and dystrophin, and at the intercalated disc interacting with SAP97. And I will show you two different mouse models that we have been generating to challenge these models. So the first one is quite straightforward. This is a model where we, in fact, knocked in, instead of the serine here, a stop codon. So the last 3 amino acids will not be expressed in the sodium channels of those mice. And when I was trying to generate that mouse, it was clear to me, at least in my head, I thought it would have been clear that a mouse that would be homozygotes with not no possibility to interact with either this PDZ, SAP97, or dystrophin, citrophin protein would not survive. I wrote that in the grant. We will most likely study the heterozygotes. We were wrong. They survived very well. I will show you only the results that we obtained with these homozygote mice where there is no SIV domain. So that's the delta SIV mouse. So what we observed was that when we looked at the expression level of NUV1.5 in this mouse by Western blotting, so we observed that there was a decrease in expression, not huge, about 25 to 30% decrease in whole cell expression of NUV1.5. So no change in the messenger of NUV1.5 and no change in any other proteins that were shown to be interacting with NUV1.5, such as syntrophin, dystrophin, utrophin, SAP97, COLEX43, and KR2.1. So consistently, we observed also a decrease of the whole cell currents. As you can see here, a decrease of about 30% of the sodium current, the whole cell sodium current, consistent with this decrease in expression, total expression of the cell. So no change in these two basic biophysical properties, activation curve and inactivation curve. So when we looked at the expression of the sodium channel in those delta SIV mouse hearts, so that's the wild type, and again here in the wild type we observe this lateral pool, which is colocalized with syntrophin, and this intercalated disc pool where there is no syntrophin. And the big surprise for us is that the intercalated disc pool does not need this SIV domain. So this interaction that we thought would be important with SAP97 at the intercalated disc, in fact, was not that important. There is still a robust expression of NUV1.5 at the intercalated disc, even though it cannot interact with SAP97 or other proteins. So the bit more striking observation was the lack or the loss or the reduction of the expression of NUV1.5 at the lateral membrane. This is very well shown here. And here on the overlap, you see that at the lateral membrane, while here it's very red, and here it's yellowish, where suggesting that there is a decrease of the lateral membrane pool of NUV1.5 here. So this is also well seen here in these isolated myocytes, where you can see here that in this merge, you have in green NUV1.5, we have the green channels at the intercalated disc, we have the channels that are at the lateral membrane, and we have most likely a third pool that is, I think, and we're currently working on that, trying to demonstrate this, a T-tubular pool of sodium channels. What you can see here is that when we mutated the last 3 amino acids, we truncated that, this is actually only the lateral membrane pool that disappeared, not the intercalated disc, not the T-tubular pool that was modified in those mice. So we performed also dualing assays with cardiac section that's shown here. And what you can see here is that we have a nice stain with these red dots of the lateral membrane pool with NUV1.5 and syntrophin in wild type. In green, this is codexin 43. This is just to show where we have the intercalated disc. And you see here that we don't have any interaction with syntrophin and NUV1.5 at the intercalated disc, because syntrophin is not expressed there. And when we have dislocations, we have no signal at all. So either because there is no sodium channels, or because the sodium channels that are there cannot interact with syntrophin because there is this truncation of the last 3 amino acids. Now a bit of functional phenotyping in that SIV mouse, and in fact it has been done with isolated myocytes, and it has been done in fact in collaboration with the group in Amsterdam, Arie Verkerk, and the group of Caron and Remy performed the experiment at that time. Now we are able to perform that also in our own lab. So he has been isolating pairs of ventricular myocytes. And when he was performing what we call macropatch recordings, means that these are not whole cell recordings. He gets the membrane patch under the pipette, and he records the current that is close to the intercalate disc. You cannot patch an intercalate disc. It's not possible. I mean, you are very close to it. Even though now there are new techniques that may be with this Gorelick technique, with this to be able to patch on the side an intercalate disc, but well, it's quite tricky. That's much more simple. So we didn't find any difference in current when he was very close to the intercalate disc. When he was in the middle of a single cell, so there was a very large decrease of current, about 60%. So very consistent with the images that I showed you before, that in fact by removing those three last amino acids, we get rid of one specific pool, which is this lateral membrane pool of NU1.5. There is still a small current here, about 30%, that is, well, only 30% that is remaining. My assumption or my hypothesis here is that this pool is the pool in the titulus that is not SIV dependent. Then we try to figure out, well, since those results were sort of contradicting our assumptions that this SIV domain was very important for the intercalate disc, we thought, well, now we will show it with the SAP97 knockout mice. And we performed SAP97 knockout mice, Kaya specific, and this has been a collaboration again with a group of Stefan and friends. And it is to show you that when we isolated myocytes here of those hearts where we have these knockouts, we have almost no SAP97 being expressed in these different isolated myocyte fractions. And this is to show you that indeed SAP97 was at the level of the tissue being reduced or knocked out in those heart sections. Those are heart sections from mice that are knocked out or controlled. And this is what I told you before. We revised a bit, at least in mice. It looks like that SAP97 is not only well expressed at intercalate disc, but there are also maybe a intracellular staining and lateral membrane staining of the SAP97. What we observed when we were looking at the action potential duration of isolated cells from SAP97 mice was a very striking prolongation of action potential duration as shown here. We looked at the DVT, there was no change. And you can see here that at APD 30, 50, and 90, we observed this decrease in action potential duration. We also observed, consistent with that, a prolongation of action potential of the QT interval in ECGs of those mice, and you can see here that the QTC interval was increased very significantly from 49 to 58 milliseconds, while the QRS duration was not, suggesting that there was no intraventricular conduction effect in those mice. So trying to understand where these action potential duration and, if you want, long QT syndromes are coming from, we looked at the different K currents. So we looked at your paper, Jen, that we're dissecting the different K current involved in the repolarization in mice, and we performed many of the protocols that you have been using. So the first K current that we looked at was IK1 current, and you can see here that there is a very strong decrease of the IK1 current. We looked at the ITO current, and again there was a very important decrease in the ITO current, but also in this IKUR current, a large significant decrease in the IKUR current. And this was extremely interesting because those tree shells were found to be also interacting with SAP97, clearly suggesting that indeed the level of expression, at least the current, is depending on the expression of SAP97. But when we looked at the sodium current, we found no difference. And here this is clearly contradicting those cellular experiments that we performed before when we acutely decrease the expression of SAP97 in HEC293 cells or in rat myocytes. So here we still don't understand whether this is some sort of in vivo rescue, or if whether there is this interaction with SAP97 is not important at all for expressing the solution consensus. So I'm going to look at the different the expression of NEB1.5 in wild type as compared to SAP97 knockout mouse. We didn't find any significant difference. When we look at the expression of the different proteins, well, first of all, NEB1.5 was not modified as shown here, but quite surprising, KR2.1, KB1.2, 4.3, and KB1.5 that are the different channels that are important for the different currents I mentioned to you, where even though those ones are quite variable, I'm not very happy about that, but at least statistically we didn't find any difference in whole cell expression of these proteins, suggesting that if there is less current, it's maybe just because the trafficking and the amount of K channels at the cell surface of the myocyte is decreased. So last, to give some sort of clinical relevance to what we have been doing, we were collaborating with, as I mentioned at the beginning, with different groups of geneticists, and in the frame of collaboration with Connie Betzina in Amsterdam, she told us, well, in fact, there is one patient. It's only one, but at least it was interesting, a patient with Brugada syndrome. Well, that's quite typical for a Brugada syndrome ECG. And this patient has, well, not had the homozygous, but had the heterozygous state, a mutation in the SIV where the three last amino acids SIV are in fact SIM, or the valine is a product mutation is mutated into a methionine. And what we performed was a pull-down experiment where, again, we changed the valine into this methionine, and we tried to pull down SAP97 and syntrophin by using this pan-syntrophin antibody. It's quite interesting, even though I'm not sure I'm able to really correctly interpret these experiments. We clearly see a decrease, a significant decrease with three different amounts of loading or incubations of lysates with this antibody against SAP97. We clearly see a decreased interaction with SAP97, while there was no decrease due to this mutation for syntrophin as shown here. When we expressed this mutant channel in HEC293 cells, what we observed first was that the surface expression was decreased by about 25%, while the whole cell expression was not. And looking at the current, we had these consistent decrease of about 25 to 30% in the channel expression in HEC293 cells. Well, consistent with suggesting that in this patient, we lost the Brugada syndrome phenotype may have been caused by this loss of expression of sodium channel in these cells. So I'd like to summarize and go through with you through a few open questions that we still have. So we have at least those two membrane complexes, the one at the lateral membrane, the one at the incatenated disc rest, now working on the idea that there may be one at the T-tubulus, not SIV dependent. We show that dystrophin and syntrophin is very important, and this SIV domain is very important for the stability of the sodium channel and the lateral membrane. And we are not yet completely sure to understand, how it works. what I can tell you is that we have made a short study by treating mice with the proteasome inhibitors for the NDX mice, but with the proteasome inhibitors that we could rescue this phenotype. It seems that at the integrated this NM1.5 may interact with SAP97, but it's not playing the role that we were thinking it is. It's not stabilizing it there. It may have another role because the results that we obtained with the delta SIV and SAP97 mouse knockout mouse model really show that this is not a stabilization or a locating problem. And obviously, we are quite interested to try to figure out what could be the specific roles of these different pools of sodium channels. You can follow us on Twitter and website, and that's the group of Mr. Bern and our collaborators in Paris and Amsterdam. I thank you very much.
I wonder what your hypotheses are for what the roles of the different populations are. I was thinking initially, like, for example, the lateral sodium channels with synchronized contraction, but now it looks like there's a big conduction problem.
Yeah, well, I forgot to maybe to show one slide, but right, we have. Was looking for colors. So we have the one at the intercalated disc, maybe a pool here, and a pool here. So with this open and this pool. So first, our assumptions based on all the different experiments that we've done, and I think this is important that people are trying to model that, and no one has tried to model it so far. So is that the sodium current that we record from a whole cell, that's 100%, and we think that about 50% of the current is originating from the integrated disc, about 20% in the teachable, and 30% here. It's most likely a naive representation of the reality, but as you know, we'll never get to the reality, so let's say naive. And yeah, this is most likely playing a role in conduction, but most likely in oriented conduction, right? These longitudinal conduction. I didn't show it here, but because it would have been too long, but I think I should have done that. In fact, when we looked at the mapping experiments in mice with the beta SIV, where this pool is reduced, we have a decrease in transversal conduction. Right. This pool is involved in longitudinal conduction, but also in transversal conduction, and maybe over proportionally in transversal conduction, because in fact the anisotropy ratio was modified by loss. This one, right now, I think it's quite wild hypothesis, but they may be very close to calcium channels, may even interact with calcium channels. I don't know, very close, and be involved in depolarization of the tubules. So, let's say conduction, longitudinal, transversal, and excitation of the tubules could be a hypothetical models for the roles. But this is under resting, state based. I think that one of the keys also is going to be to look at differential regulations of the differences.
So several comments maybe or insights from modeling also related. So one, the lack of sensitivity of phenotype to genotype, or the reduction of sodium channels, part of it is because sodium channels are expressed with a huge reserve in cardiac myocytes. And you can go in a single cell down to 11%.
Yeah, I don't think so.
Yeah.
No.
You can go down to 11% of expression level to still get an action potential. So if you reduce it by 30%, you might not see an effect. That's one. The other one is that Jan Kuchera, when he was in my lab, Jan Kuchera is now in Bern, if you don't know, but he was.
I'm meeting with him
When
almost every day.
When Jan was in my lab, he simulated different proportions of sodium channels in different parts of the cell. And he showed that there is no difference whether they are distributed uniformly around the cell or they are concentrated, all of them, in the intercalated disc, unless you have very high degree of uncoupling at the gap change. So for well-coupled cells, or even some reduced coupling, we didn't see any effect of the distribution and where they are. The third thing that comes to mind is for the T-tubules. I thought that Bill Catterall showed the other type, NAV.
Yeah, well, he showed he had TTX sensitive neuronal isoforms. He couldn't say which one because it was just a figure. I can tell you, I doubt about. I mean, I doubt about that. I'll tell you one thing. This is also something related to your work and with Celine. In mouse myocytes, right? We didn't do those experiments with other mice. If you look at the RNAs, the messengers, if you do RNA-seq of isolated myocytes, only isolated myocytes, you see only 2 sodium channel genes, SCN5A, and the one that is about 10 times less expressed is SCN4A, and the others are not expressed. We don't see them. Right? And if you look at the experiment of Celine, when she pulled down using her pan sodium channel antibody, she found only, and about in the same proportion, so 10 times more SCN5A, peptides for 4A. I think this is the other channel. That is
your assumption about the T-tube is that it helps to depolarize them fast, so you have more synchrony of calcium release.
Right.
That's the idea.
Right. And also, I think that the regulations here could be a bit similar. We were trying to figure out whether PK would also target this specific group. Don't have now yet exactly.
There's also sodium calcium exchanger there with higher expression.
Yeah.
So
Yeah, right. So the cell, the cardiac cell, well, as you know, it's very compartmentalized, very well polarized, and our evidence suggesting that we have different roles of sodium channels.
Have you done the acute knockdown of SAT97 in the mouse?
Right. That's what Anobor Justus in Khalifa's lab has done. We didn't do that, and he observed very, very similar findings that we did. So very acutely down regulation or down expression of K currents, and the sodium current is increased. And we also had something similar actually. In fact, when we look at the statistic of expression of sodium channel, we had also a small increase in amount of proteins. We didn't know what to do with it, but he has very similar results.
So given that mutations in the same gene are associated with many different clinical phenotypes, what do you think, are they on a spectrum of severity? Are they gain and loss of function? Is anything
I think it has to do with the different roles that we don't know yet exactly, and different roles in a given cell, but different roles in different tissue types, in contextual tissue, in ventricles, in epicardium, in atrial. I think, well, the role of the sodium channel is more subtle, more multiple, more roles than.
Is there anything known about the particular domains that are associated with more atrial, for example, versus ventricular phenotypes?
Yeah, there have been some hints about, for instance, it looks like that mutations that are found in the S4 domains may lead to a dilated cardiomyopathy, for instance. that there are a few things like this.
So how do you think about all this? Do you think that all of these proteins are like SAT97, for example, now there's 4 channels at least that it interacts with. So does that mean that there's 4 different sites of interaction and one molecule is interacting with lots of different channel types? Or is there a single or...
Yeah.
Is there like 5 times as much SAT97 as everybody else? Or?
Naively, I would say, my first answer would be yes. I mean, there are as many SAT97 as K
So you
channels.
think they're always associated? It's not like a dynamic interaction that they're coming apart?
Possibility is there. It may not be always. And for instance, for SAT97, I just looked at that paper recently with one of my students. I mean, it has been shown that in one of those K channels, I think it was KR, the interaction, and this was Carol Vandenberg, was shown that depends on the phosphorylation of one of the serine residues, and it's PKA regulated. So I think that there may be regulation and the dynamicity in these interactions. Sure.
I guess
Any other questions, comments? Anybody wants to go there?
Now we are looking for PhD students, right? That's a good one. We're looking for a PhD student. Any
All right, thank you very much.